Hello, seventh graders. We are about to start a new unit. It is a unit on genetics, and it happens to be one of my favorite for the whole year. I find the whole topic fascinating. We're going to spend quite a bit of time on it because we're going to start with a unit on basic genetics, understanding how traits are passed from one generation to the next. And then we're going to do a unit called modern genetics, where we talk about how scientists, researchers have taken the whole idea of DNA and genes and what we've learned recently in order to make advancements to help people, to help society. So it's going to be a great couple of weeks. I'm looking forward to it. Let's start with the basic definition. So genetics is the science of heredity, how traits, characteristics get passed down from one generation to the next. Why is it that you have your mother's hair but your father's eyes? Maybe you happen to look exactly like one parent rather than the other one. Why is that so? These are the questions that we're going to be exploring in this unit.
You are part of a diverse array of organisms that inhabit the biosphere on Earth. Bacteria, plants, animals, and you are all carbon and water-based cellular forms with complex organization and heritable genetic information. So, are you curious what makes you, you? Your genes make you, you. Genes make up the set of instructions that are given to you from your mother and your father. These instructions are in almost every cell of every living thing and are stored on extremely long strands of chemicals called DNA. DNA stands for deoxyribonucleic acid, and these long strands are called chromosomes. Chromosomes are made up of many genes. One gene is a specific sequence of DNA on a chromosome that provides particular cellular instruction. DNA is shaped like a ladder that's been twisted, which is called a double helix. The steps of the ladder are made of four main chemicals called bases: adenine, thymine, guanine, and cytosine. The particular order of A's, T's, G's, and C's is extremely important as it determines whether you are a banana, rhinoceros, or a human. It also determines whether you have brown hair, blue eyes, or green skin. The sides of the ladder are made of alternating sugar and phosphate molecules. The entire DNA sequence is called a genome, and in humans is 3.2 billion base pairs. This would take 9.5 years to read out loud without stopping. It would take someone typing at 10 words per minute for 8 hours a day, 50 years to type the entire human genome. If you unwrap your DNA from one cell, it would extend more than 6 feet long. If you unwrap all of the DNA in all of your cells, it would reach the moon 6,000 times. All 3 billion bases of your genome would take 3 gigabytes of computer storage space. Your entire DNA sequence would fill 200 New York City telephone directories. You are 99.9% identical to the person next to you. Only 0.1% of our genetic makeup differs. Less than 2% of your DNA carries recognizable instructions to make proteins. The rest is called junk DNA, as it does not currently appear to code for anything. It takes around 8 hours for one of your cells to completely copy its DNA sequence. You are you because of your genes. Now you know more about this microscopic entity that makes us all the same, but grants you with distinct differences that make you unique.
So we've already done some of the legwork for this unit because we've learned about DNA in a prior unit. What we know about DNA and its structure and the nitrogen bases and the sugar and the phosphates is relatively new information. Only within the last maybe 100 years have we known about that. The beginnings of genetics, the beginnings of trying to understand how traits get passed down from one generation to the next, actually started with a man called Gregor Mendel. He's actually known as the father of genetics. He lived in the 1800s, so a long time before we even knew anything about DNA. He was an Austrian monk. He was a religious man. He lived in a seminary with other religious men. And being a religious man, he was fairly educated and curious. And one of his jobs at the seminary was to work in the garden. The seminary needed to provide their own food for themselves. So they had a large, extensive garden, and Gregor Mendel worked in the garden. And he began to notice that, in particular with pea plants, that there seemed to be certain traits that would appear in some generations and maybe not another generation. So for example, pea plants have purple or white flowers, and there seem to be certain generations where the purple flowers seem to be quite evident, and then other generations where the white flowers were evident. And he kept kind of noticing this and wondering about why that was so, why you wouldn't have an equal number of purple flowers and white flowers. So he started experimenting with pea plants, and pea plants are not the most exciting thing to experiment with. Certainly animals or people might be more interesting, but some of the advantages to the pea plants were that they have a really short life cycle. So you can grow a pea plant from seed to maturity in just a matter of weeks. And if Mendel was interested in looking at patterns of inheritance, how traits are passed down from one generation to the next, he needed to see lots of generations. So within a year, he could see maybe, 8 to 10 generations of pea plants. So he'd really be able to see how a trait was passed from generation 1 to generation 2 to generation 3, etc. Another reason why pea plants were good to study is because they have very visible traits. You can look at a pea plant and very clearly see if it is tall or short, if it has purple flowers or white flowers, green seeds or yellow seeds, wrinkle seeds or smooth seeds. So Mendel could, quickly kind of make his observations. And then finally, for many of the traits that pea plants have, there are only 2 versions. So it's not like the flowers can be purple, white, yellow, pink, and red. The flowers are only purple or white. The size is only tall or short. The seeds are either green or yellow. So having just 2 options made it easier for him to track the traits through the different generations. Even though he was working in the 1800s and he had no access to technology, his experiments were really successful. So successful that we still rely on them today. The basic information that we learned from his pea plants, we still use today. One of the reasons he was so successful is that he kept great records. So he had lots of notebooks and journals, and he would make observations of the first generation, lots of different characteristics in the first generation, and then he would record the next generation's traits and the next generation's traits so that he could go back and look at patterns. In the beginning of the year, when we talked about what makes a good scientific experiment, we said that it's important to have a lot of samples. It's not enough to just test one pea plant. Whatever results you get from one pea plant, people are not going to have faith in. People are not going to think of it as valid. It could just be a fluke. But if you had 100 samples, 1,000 samples, and you still consistently kept getting the same results, people would have more confidence in that. So Mendel definitely had hundreds and hundreds of pea plants. He also tried to keep it simple. He focused on one trait at a time. He wasn't looking for combinations of purple flowers, tall pea plants, and wrinkled seeds. He just looked at height and tried to see the pattern in the various generations. He just looked at flower color and tried to see the patterns that existed. So Mendel cross-pollinated tall plants with short plants. So here's our opportunity to review what cross-pollination means. So we learned in our plant unit that pollination is when the pollen from this stamen is transferred to the pistil and leads to the process of fertilization, which will produce a seed. Cross-pollination is when the pollen from the stamen travels to the pistil of a different plant, maybe a neighboring plant, but not the same exact plant. Self-pollination is when the pollen from the stamen travels to the pistil of the same exact plant. So Mendel acted as the pollinator. He didn't rely on insects. He didn't rely on the wind because he wanted to be the one in control of where the pollen came from. He wanted to know which two pea plants were pollinating each other so that he could see what result that had on the offspring. So he took a brush or a Q-tip or something like that, and he actually physically took the pollen from the stamen and he brushed it on the pistil of whatever plant he wanted to mate with his initial plant. So he crossed tall plants with short plants, and the result was all tall plants. This kind of surprised Mendel. He would have thought that he would have gotten some tall plants and some short plants, but he didn't. The whole entire new generation was tall plants. So his curiosity kept him experimenting. So he took those tall offspring that he had just made and he mated them together. He pollinated them together. So now he crossed his tall offspring with this tall offspring. And this time he got mostly tall plants, but some short plants. Now this confused him. Again, he was confused. He thought, that if he crossed a tall plant with a tall plant, he would get all tall offspring, but that was not the case. He continued his work for generations and generations, and he looked at other traits, and he kept seeing the same pattern. So after years of looking at his results, looking at his data, he came up with a couple of conclusions, a couple of findings. His first finding was that factors that control traits exist in pairs. In other words, pea plants have two factors that control what color their flowers are going to be. Pea plants have two factors that control whether they're going to be short or tall. At the time, he just called them factors. Today, we call them genes, but that was not a word that was used in Mendel's time. Another conclusion he came up with was that of these two factors, one of them is contributed to by the mother, and the other is contributed from the father. So a pea plant that is tall is going to have two factors for height. One of those factors is going to come from the mother plant. That seems kind of weird. And the other factor is going to come from the father plant. And then finally, his last conclusion was that if you have two factors for a trait, but they both don't show up, then that must mean that one of these factors is hiding or masking the other one. That if you have two factors present, they're not potentially both going to show up. We eventually called these factors the dominant factor and the recessive factor. So the dominant factor is the factor that is going to mask the other factor, mask the recessive factor. If the dominant factor is present, it is going to show up. It is going to be expressed. The recessive factor is the one that is going to be hidden. If it is present with the dominant factor is going to overshadow it, and it's not going to show up. The only way a recessive factor would show up is if the recessive factor is the only gene, the only factor present. So, a person, an organism, has two factors for every trait. So here are the three possibilities, three possible combinations that a person or an organism could have. A person or an organism could have two dominant factors, two dominant traits. And if that's the case, that means that they are actually going to have the dominant trait. When we look at that person, look at that organism, the physical characteristic that we see is going to be that of the dominant trait. That's the only possibility, right? If a person or an organism only has two factors and they're both dominant factors, then clearly the only trait that we're going to see is the dominant trait. Another possible combination is that a person could have two recessive factors. If a person or an organism has two recessive factors, they're going to have the recessive trait. That's the trait that we're going to see when we look at them. That's the only possibility they can have. They only have recessive factors, so that is the factor that's going to show up. And then our third possibility is that a person could have a dominant factor and a recessive factor. Well, if they have a combination of a dominant and a recessive factor, that dominant factor is going to hide the recessive factor. So the only factor that we're going to be able to see is going to be the dominant factor, the dominant trait. The organism is going to express or have the dominant trait. So let's review Mendel's experiments to see how this works. At first, he had a tall pea plant and a short pea plant, and he cross-pollinated them. So the tall pea plant must have contributed a tall factor or gene. The short pea plant must have contributed a short factor or a short gene. The tall was dominant to the short. The tall was going to hide the short factor, the short gene. So all the pea plants in that first generation were tall. But now we moved on to Mendel's second generation. So he crossed the tall pea plants with the tall pea plants, but those tall pea plants each had two factors for height. They each had a dominant factor and a recessive factor, which meant that depending on which factor the pea plant passed along, that new offspring could have two dominant factors, so they would be tall. They could have a dominant and a recessive, which means they still would be tall. Or they could have two recessives, which means they would be short. And that's why the next generation in Mendel's experiment had mostly tall, but some short pea plants. As we move forward in this genetics unit, we are going to be using capital letters, uppercase letters, to represent the dominant genes. And we are going to be using lowercase letters to represent the recessive genes. That makes sense. The dominant genes are the ones that are going to show up. They're going to be the ones that are going to hide or mask the recessive genes. So it makes sense that we use uppercase for dominant and lowercase for recessive. Some vocabulary that we'll be using going forward. If an organism has two of the same genes, either two dominant or two recessive, we call that organism pure or homozygous. They mean the same thing. We can use them interchangeably. That prefix homo means same. So a person or an organism in this case would have two dominant or two recessive. So I just mentioned that we're going to use letters to represent dominant and recessive factors. So here we have two uppercase T, so this would be two dominant genes for height, for tallness, and then two lowercase genes representing the recessive. Some more vocabulary. If we have two different genes, if an organism has a dominant and a recessive, we call that organism a hybrid or a heterozygous, hetero meaning different. Again, if we're going to use those letters, if you're going to have one dominant factor and one recessive factor, you're going to have an uppercase T and a lowercase t.
These days, scientists know how you inherit characteristics from your parents. They're able to calculate probabilities of having a specific trait or getting a genetic disease according to the information they have from the parents and the family history. But how is this possible? To understand how traits pass from one living being to its descendants, we need to go back in time to the 19th century and a man named Gregor Mendel. Mendel was an Austrian monk and biologist who loved to work with plants.
By breeding the pea plants he was growing in the monastery's garden, he discovered the principles that rule heredity. In one of the most classic examples, Mendel combined a purebred yellow-seeded plant with a purebred green-seeded plant, and he got only yellow seeds. He called the yellow color trait the dominant one, because it was expressed in all the new seeds. Then he let the new yellow-seeded hybrid plants self-fertilize, and in this second generation he got both yellow and green seeds, which meant that the green trait had been hidden by the dominant yellow. He called this hidden trait the recessive trait. From those results, Mendel inferred that each trait depends on a pair of factors, one of them coming from the mother and the other from the father. Now we know that these factors are called alleles and represent the different variations of a gene. Depending on which type of allele Mendel found in each seed, we can have what we call a homozygous pea, where both alleles are identical, and what we call a heterozygous pea, when the two alleles are different. This combination of alleles is known as genotype, and its result, being yellow or green, is called phenotype. To clearly visualize how alleles are distributed amongst descendants, we can use a diagram called the Punnett square. You just place the different alleles on both axes, and then you figure out the possible combinations. Let's look at Mendel's peas, for example. Let's write the dominant yellow allele as an uppercase Y, and the recessive green allele as a lowercase y. The uppercase y always overpowers its lowercase friend, so the only time you get green babies is if you have two lowercase y's. In Mendel's first generation, the yellow homozygous pea mom will give each pea kid a yellow dominant allele, and the green homozygous pea dad will give a green recessive allele, so all the pea kids will be yellow heterozygous. Then, in the second generation, where the two heterozygous kids marry, their babies could have any of the three possible genotypes, showing the two possible phenotypes in a 3-to-1 proportion. But even peas have a lot of characteristics. For example, besides being yellow or green, peas may be round or wrinkled. So we could have all these possible combinations: round yellow peas, round green peas, wrinkled yellow peas, and wrinkled green peas. To calculate the proportions for each genotype and phenotype, we can use a Punnett square too. Of course, this will make it a little more complex. And lots of things are more complicated than peas, like, say, people. These days, scientists know a lot more about genetics and heredity, and there are many other ways in which some characteristics are inherited, but it all started with Mendel and his peas.
There you have it. There's our introduction to genetics. I look forward to exploring it with you over the next couple of weeks.